Here are the answers to today's review sheet. First, looking at DNA and RNA, you were supposed to complete the following table to illustrate the differences. Hopefully you could have found those from what looked like a T table in your notes. The sugar of DNA is deoxyribose. The sugar of RNA is ribose. The nitrogen bases in DNA are A, T, C, and G. In RNA, A, U, C, and G. DNA has 2 strands and is a double helix. And RNA is 1 strand and it does not have a helix, or you could just have written single-stranded. The location of DNA is in the nucleus, and the location of RNA is cytoplasm. The function of DNA is to store information, and the function of RNA is to transmit information. Moving on to the compare and contrast chart between DNA and RNA, both of them are made of nucleotides. Only DNA has deoxyribose, only RNA has ribose. RNA is single-stranded, DNA is double-stranded. Both do contain the nitrogenous bases. Only DNA has thymine, whereas RNA has uracil. DNA is the one that serves as a template for the synthesis of mRNA, which is a nucleic acid. DNA is the double helix. Replication is the process of copying DNA. Transcription is the process of making RNA. DNA is going to make an exact copy during replication, whereas RNA is going to be the messenger. There are 3 forms of RNA, tRNA, mRNA, and rRNA, so that's the one that has more than one form. DNA is found in the nucleus, RNA leaves the nucleus for the cytoplasm, and DNA does not leave the nucleus. So there's your comparison and contrast table. Moving on to the process of protein synthesis, hopefully you remember at the beginning of this chapter we said that transcription and translation is the process of using the information stored in DNA to create a protein. So DNA is located in the nucleus of a cell. It specifies the production of protein out in the on a ribosome in the cytoplasm. RNA contains the sugar ribose, as we said above, and has uracil instead of thymine, and lacks a helix and is single-stranded. mRNA carries the coded sequence of bases to the cytoplasm. If you were looking to be very specific, so messenger RNA carries the coded sequence of bases to the cytoplasm, and if you were looking to be very specific, you would have said ribosome, because the ribosomes, of course, are located in the cytoplasm. Ribosomal RNA, or some of you might have written simply lowercase r, RNA, is found in the ribosomes, and transfer, or tRNA, carries amino acids to the ribosomes. The latest hypothesis is that one gene controls the synthesis of one protein, but we said in class that's not entirely true. We think it's actually a bit more complicated than that. A gene is a segment of DNA that specifies the sequence of amino acids and a polypeptide of a protein. So remember, we said that a gene is a stretch of DNA that codes for a specific protein. Transcription occurs in the nucleus when DNA acts as a template for M, or messenger RNA synthesis. Translation occurs in the cytoplasm when the sequence of mRNA codons determines the sequence of amino acids in a polypeptide. Remember, the goal is to take the information stored in DNA, turn it into a readable message of mRNA, and then translate that message from the language of nucleic acid to the language of amino acid, which just means make proteins. The nucleotide code for 20 amino acids is a triplet code. Each 3-letter unit of an mRNA molecule that codes for a particular amino acid is called a codon. The codon for methionine, which is AUG, is the codon in reference, signals polypeptide initiation. Remember, you don't need to memorize that. You're going to be given the genetic code chart. During transcription, the DNA helix unwinds, unzips, and complementary RNA nucleotides pair with DNA nucleotides on one strand, the template strand. So this is the beginning of protein synthesis with the beginning of transcription right there. RNA nucleotides are joined by RNA polymerase. Remember, it's an enzyme if it ends in ase. Wherever A, T, G, or C is present in the DNA, A, remember, pairs with which one of our other nitrogen bases? A always pairs with U, T always pairs with A, G always pairs with C, and C always pairs with G. And that's incorporated into the mRNA. The next question is asking you about gene editing or splicing. And remember, we said that there are 2 sections of that template that's going to be, there are 2 different sections. One section will be used and one's going to be edited out. The parts that are included are called exons. Remember, X marks the spot. Introns are the ones that are edited out. So introns are portions of DNA that are not part of the gene product. They get edited out, whereas X marks the spot, exons are portions of the gene that are expressed to form a protein product. The sequence of codons in mRNA specifies the order of amino acids in a polypeptide. The next question is just asking you what that T and tRNA stands for, and you should know it stands for transfer. It brings amino acids to the ribosome in the cytoplasm. tRNA molecules have an amino acid attached to one end and the an kind of gives us a hint, anti-codon at the other end. During the initiation step of translation, mRNA binds to the ribosome. During elongation, the polypeptide lengthens one amino acid at a time, and during termination, we encounter a stop codon. So the next set of instructions is asking you to match a term to the event that occurs during that term or to the vocabulary word to which it refers. The process listed above that creates protein is translation. That creates RNA is transcription. It occurs inside the nucleus, that's transcription again. Utilizes a stop codon on mRNA. Well, that's specifically, it's translation, but what part of translation? J, the termination step. The process occurs in the cytoplasm of the cell. That's going to be translation. The tRNA amino acid complex binds to the ribosome. That happens during initiation of translation. Initiation, elongation, and termination steps, those are all actually in both A and B. Carries information for polypeptide synthesis. Well, we know the carrier molecule, the messenger molecule, is mRNA. Found on tRNA, and it binds to mRNA. That's the anticodon. mRNA binds to 2 ribosomal subunits. That's also initiation of translation. And the sequence of 3 RNA nucleotides that code for a particular amino acid, that's a codon. Since I'm using my iPad to record this answer key for you guys, I don't have a codon table in front of me. But I do want to remind you, as you look at this genetic code, or as you look at this chart, to use your genetic code. And remember, when you're determining the amino acid, you're going to use the codon. Always use the genetic code with the mRNA codon. If you don't, you'll end up with all the wrong answers. Remember, when you are making the complementary RNA to any DNA, to replace any instance that you would find a thymine, replace it with a uracil. So in the first one, AAA should be UUU. The tRNA anticodon is AAA, and you would use UUU for the amino acid. Methionine, we know, is AUG for our anticodon, so that's a TAC and a UAC for our anticodon. And you should be able to match the rest, remembering that this you need the genetic code chart for. The more important question on this page for me is the elongation stage of translation. I'm going to use this space to do a brief review of all of transcription and translation, and then I'll point out what would be just the answer to this particular question. So, remember, we start with our central dogma of biology. DNA codes for RNA, which codes for protein. The process of making DNA into RNA is called transcription. That process creates an mRNA molecule. This happens in the nucleus, where the mRNA simply is read to make a protein in the process of translation. Our mRNA molecule is the result of complementary base pairing with a template region of DNA. That's read from 3' to 5' on the template, so the mRNA is built from 5 to 3. It gets edited where exons are kept and introns are edited out, a cap and tail are added, and then the mRNA can leave the nucleus to move to a ribosome. Once that happens, and we've moved to our ribosome, we have our cap and our tail on an mRNA molecule. Initiation happens when a small ribosomal subunit and a large ribosomal subunit come together. And remember, on our ribosome we have 3 spots. We have the exit, we have the spot that protein is made, and we have the entrance. We read 3 mRNA bases at a time in the form of a codon. Let me change colors here, use this one. A transfer RNA is going to bind to the first codon right here with its anticodon, that would be right there at the bottom. And at the top of that is an amino acid. This is initiation. We move on to elongation when another tRNA molecule slides into the A spot. It's matching with its anticodon to the codon that occurs on the mRNA below it, and it brings a matching amino acid. Elongation starts when the mRNA slides to the left and the initial tRNA enters the exit site, the one that's in the A site enters the P site, and then another one can come enter the A site. Then you have elongation. Termination happens when the A site is occupied by a stop codon, of which there are 3 choices. That should be all of the answers on your review sheet. If you have any questions, you know where to find me in the morning before school. And I think that should cover it. I'll see you guys tomorrow.